Welcome to class number 5, session number 1. This class will deal with sensing devices that are based on quantum dots. In this session, I will first provide a definition of the quantum dots as well as the main properties of the quantum dots. Quantum dots are small semiconducting particles with typical sizes that range from 1 nanometer to 10 nanometer. Quantum dots are artificial clusters of semiconductive atoms that have the ability to confine the electron's motion due to their small size. One of the most important properties of the quantum dots is the ability to tune their band gap and therefore to control the light absorbance and emission frequencies. In this way, it is possible for their optical and electrical properties to be adjusted according to their purpose of use. Quantum dots absorb photons of light and then re-emit longer wavelength photons for a period of time. The high controllability level over the quantum dot size provides very precise control over the wavelength of the re-emitted photon. That means that the color of the light emitted from the quantum dot can actually be manipulated without significant cost or without the use of high-end technology. Following this procedure, a full range of quantum dots can be manufactured, each with a narrow, distinct emission spectrum. Now, besides confinement in all 3 dimensions, namely in the case of quantum dots, one can find other quantum-confined semiconductors, and those include quantum wires, which confine electrons or holes in 2 spatial dimensions and allow free propagation in the 3rd dimension. Also, one can find quantum wells, which confine electrons or holes in 1 dimension and allow free propagation in the rest 2 dimensions. To understand the mechanism behind the quantum dots, we have to recall first that electrons exist in discrete energy levels in the bulk semiconductor. In these materials, there is a forbidden range of energy levels, most known as band gap. The lower energy level is called the valence band, and the higher energy level, namely that above the energy gap, is called the conduction band. By absorbing some sort of stimulus, such as light or heat form, an electron can rise to the conduction band from the valence band. This action leaves behind a hole in the valence band, and the hole and the electron together are called an exciton. The average distance between the electron and the hole in an exciton is called the excited Bohr radius. When the size of the semiconductor falls below the Bohr radius, the semiconductor is called a quantum dot. The exciton have a limited lifetime, and eventually the electron will come back to the valence band and recombine with the hole. The recombination process is usually a radiated process, which includes a photon release, or, in other words, a fluorescence. The absorption and the fluorescence processes can be measured in this case, and also it is noteworthy to point out in this context that the confinement in the quantum dots can also arise from electrostatic potentials, usually generated from or by external electrodes, doping, strain, or impurities. Generally, the smaller the size of the crystal, the larger the band gap, the greater the difference in the energy between the highest valence band and the lowest conduction band becomes. Therefore, more energy is needed to excite the dot, and simultaneously more energy is released when the crystal returns to its resting state. This equates to higher frequencies of light emitted after excitation of the dot as the crystal size grows smaller, resulting in a color shift from red to blue in the light emitted. In addition to such tuning, a main advantage with the quantum dots is that, because of the high level of the control possible of their size, it is possible to have a very precise control over their conductive properties and over, of course, the conducting material. Generally speaking, quantum dots are made largely from elements in the second and sixth group of the period system, which include, for example, cadmium or zinc, or from the third and the fifth group, which include, among the rest, phosphoids and indium arsenides. Quantum dots can be engineered to fluorescence in different wavelengths based on their physical dimensions. By use of different colloidal quantum dots for the different parts of the visible spectrum, the entire range of the spectrum can be synthesized, as seen in the current example on the current slide. The ability to functionalize quantum dots has the potential to change their properties, including solubility of the quantum dots in a variety of solvents. Also, it has the power to affect the ability of the quantum dots to specifically bind to the targets. Few examples of functionalized quantum dots include quantum dots coated with monolayer of hydrophobic thiols, which are generally stabilized ionically in a solution. Another example is the quantum dots coated with cross-linked silica shell, which in turn can be readily modified with a variety of organic functionalities using well-developed silane chemistry. Further examples include quantum dots that are encapsulated with or in amphiphilic polymer, which form highly stable or micelle-like structures. In this case, any of these quantum dots may be modified to contain polyethylene glycol, or PEG, to decrease surface charge and increase colloidal stability. Water-soluble quantum dots may be covalently or electrostatically bound to a wide range of biologically active molecules to render specificity to the biological target. Few examples include quantum dots that are conjugated to antibodies, which can yield specificity for a variety of antigens and are often prepared throughout the reaction between reduced antibody fragments with melamine PEG-activated quantum dots. Also, this include quantum dots cross-linked to small molecule ligands, inhibitors, peptides, or aptamers that can bind with a high specificity to many different cellular receptors and targets. Furthermore, quantum dots conjugated to cationic peptides, such as HIV peptide, can quickly associate with cells and become internalized via electrostatics. Quantum dots have been used to detect the presence of biomolecules using probe design incorporating energy donors or energy acceptors. For example, quantum dots can be adapted to sense the presence of the sugar maltose by conjugating the maltose binding protein to the nanocrystal surface, as seen in the left image on the screen. By initially incubating the quantum dots with an energy-accepting dye that is conjugated to a sugar recognized by the receptor, blue excitations of the quantum dots yield little fluorescence, as the energy is non-radiatively transferred to the dye, and this can be seen by the gray color in the schematics. Upon addition of maltose, the quencher sugar conjugate is displaced, restoring fluorescence in a concentration-dependent manner, and this is seen in the green color in the schematics. Quantum dots can also be sensors for specific DNA sequencing. This could be achieved by, for example, mixing the targeted single-stranded DNA with an acceptor fluorophore conjugated to DNA fragment that is complementary to one end of the target DNA. In other instances, the DNA detection could be achieved by mixing the targeted single-stranded DNA with a biophylated DNA fragment complementary to the opposite end of the target DNA. In this case, the DNA hybridized to yield a biotin DNA fluorophore conjugate. Then, upon mixing this conjugate with quantum dots, a green fluorescence is quenched throughout non-radiative energy transfer to the fluorophore conjugate, and this is seen again here in the gray color on the schematics. This dye acceptor then becomes fluorescent, thus indicating the presence of the targeted DNA. This could be seen in the red color in the middle image. Finally, quantum dots conjugated to the Lorphase enzyme can non-radiatively accept energy from the enzymatic bioluminescent oxidization on the quantum dot surface, thus exciting the quantum dots without the need for any external illumination. The use and the application of the quantum dots have many advantages. The first advantage is that the recent advances have shown that quantum dots can really make valuable contributions to a wide range of applications, thus substituting many of the bulk, expensive, and inefficient materials. The second advantage relates to the fact that quantum dots formation absorb photons of light and then re-emit longer wavelength photons for a period of time, and that the high controllability level of the size of the quantum dot provides very precise control over the wavelength of the emitted photon. That means that the color of the light emitted from the quantum dot can actually be manipulated without significant cost or without the use of high-end technology, as mentioned in the previous slides. Following this procedure, a full range of quantum dots can be manufactured, each with a narrow, distinct emission spectrum. The third advantage of the quantum dots relates to the fact that they only require a small amount of energy in order to be excited, and this can be achieved by a single blue or ultraviolet wavelength beam, regardless of the quantum dot size. Both attributes reduce the cost of the sensing quite dramatically. The fourth advantage of the quantum dots relates to their high photostability and to their brightness. This property makes them suitable for high sensitivity applications like fluorescent tagging and leaf cell imaging. Their fluorescence properties and their high resistance to metabolic degradation enable a wide range of experiments to be performed, thus ignoring possible time barriers. The fifth advantage is that the quantum dots can be used in various forms. For example, as small as crystals in liquid solutions, as quantum dust in bead form. All these existing forms make their range of applications even wider. Another great advantage of the quantum dots relates to the availability of multiple methods to develop them easily and cost-effectively. These methods include lithographic techniques, epitaxial techniques, and colloidal synthesis, as we'll see in the second session of this class. We come now to the end of class number 5, session number 1. Thank you.